This is Dr. Carey Mullis, who was the one who discovered this technique. Polymerase chain reaction is a very, exquisite example of combining DNA replication with nucleic acid hybridization to amplify a piece of DNA. What PCR is to wet lab, blast is to dry lab. So tomorrow we talk of blast, today we talk of PCR. The reagents required, you need a template DNA. So there is a larger DNA of which a sub part you're going to amplify. Most commonly, let's say you take human genomic DNA and you're amplifying a specific gap gene or let's say histone gene. And this template DNA, the concentration is around 50 to 100 nanograms per microliter of the reaction. You have a reaction buffer, which is provided by the company itself, plus HCl, magnesium ions, bovine serum albumin, and so on and so forth, right? Then the cofactor for Taq polymerase is magnesium, so therefore magnesium chloride has to be there. Then, of course, as the DNA polymerase moves, after it is initially primed, it will basically add complementary nucleotides to the growing template, and therefore it requires DNTPs. You have equimolar ratios of DNTPs of the 4 DNTPs, A, T, G, and C. You require primers because DNA polymerase cannot synthesize de novo. So DNA polymerase cannot synthesize de novo. It needs a pre-existing strand to the 3' end of which it can keep adding nucleotides, right? So therefore, you need a primer that is already hybridized to your template in the region where you want your amplification to happen. And then, of course, you have DNA polymerase, which is typically here, because here you need several cycles of denaturation and then again annealing and then again extension. So this DNA polymerase that we use here is temperature stable, and that is where you have the advent of Taq polymerase or Thermophilus aquaticus polymerase, which is coming from a bacteria which is found in hot springs. Then, of course, you have double distilled water to make up the reaction volume. So you make up the reaction up to 20 to 25 microliters as per your requirements. Now, what is happening here? So here is your larger DNA, and in this larger DNA, you want to amplify this region that is marked by the boundary A and boundary B, right? So what you do is you have to basically, ideally what you have to do is to design primers here. So you have a forward primer, which has exactly the same sequence as the top strand. And actually, when it goes to annealing, it is going to anneal to its complementary strand here, right? So, and likewise, you have a reverse primer, which is basically towards the end of boundary B. And this again is basically going to bind to the red strand on the top. So it has the same sequence as this bottom strand. And obviously, it is going to bind to the complementary strand. So it comes and binds here. So when you place your primers for your PCR for the first cycle after the annealing of the primers, this is the placement of the primers. Here is your forward primer on the bottom strand at its location of hybridization. Likewise, the reverse primer on the top strand at its location of hybridization. And then when you start your reaction, the DNA polymerase is going to add complementary nucleotides at the 3' end. This is the arrowhead is the 3' end of the primer here. Likewise, at the 3' end of the primer here, right? So both strands are extended simultaneously, but here in this case, I show you one by one. So here you have your forward primer. Likewise, you have the reverse primer getting extended, right? And this is how it works out. So after the first cycle, you have now 2 double-stranded DNA if you started with 1 molecule of double-stranded DNA. So this was your start molecule. You denatured it. You allowed the primers to hybridize, and then the primers have been extended. And after extension, now you have 2 double-stranded DNA molecules, right? Then we come to the second cycle. In the second cycle, you denature these again. So you have your denatured strands. And then again, you have the primer placement. So primers go and find their complementary regions on the DNA and hybridize. Here you are. And then you have the extension again. So here you are and here you are. Right? So this is your extension. Everybody with me, class?
Yes, sir.
Yes, sir.
Okay. Right. So this is where you are. You have your primers annealed, and then again your extensions, and this extends here, and that is how you have your second cycle. Likewise, you can keep doing this, and every cycle, the number of double-stranded DNA is going to double up from the previous cycle. That's the whole idea. So, for example, here you start with 1 molecule of DNA. After cycle 1, you have 2 molecules of double-stranded DNA. After cycle 2, you have 4 molecules. After cycle 3, you have 8 molecules. And therefore, after n cycles, you have 2 raised power n, right? So this is what it is. So cycle number 0, if the starting DNA molecule is 1, you have 2 raised power 0 is 1, right? So 2, 4, 8, 16, 32. So it follows the series of 2 raised power n. So this is when you're starting with one molecule of DNA. If you start with X molecules of DNA, then you have X into 2 raised power N molecules. That is why it is known as exponential amplification, because every cycle the amount of DNA doubles up, right? So here is a question. If X number of DNA molecules are found in N cycles, in how many cycles would the number of molecules be 2X? Anyone?
So that n plus 1 cycles.
Yes. So it will be at N plus 1 cycle, right? So the next cycle it is going to double up. So N plus 1 is correct. So this is typically your PCR plate. If you're putting up 98 samples, you can directly put up in a sequencing plate here. This has 8 rows and 12 columns, and likewise, correspondingly, your PCR machine would have as many 96 wells, right? We are talking of the various ways in which the primer can arrange, right? So you have specific annealing, which means that the primer hybridizes to absolute complementarity on the template DNA. So there is no mismatch between the primer nucleotide and the complementary nucleotide on the template, right? So this is absolutely specific annealing. You could have a bit of non-specific annealing where the primer head and tail are completely hybridized. In between, you could have, let's say, the primer has A here and here it is a C. So A and C are non-complementary nucleotides, but because it is hybridized at the tail and also at the head, this primer would also work, right? So can be used to introduce mutations in coding sequence. And sometimes when you want to introduce mutations in coding sequence to test whether the mutation results in some downstream effect or not, you use what is known as slightly, erroneous primers, so that you are deliberately introducing some error in the template DNA or nucleotides that are not present in the template, but you want them for an experimentation, right? Then you could also have something called for non-specific annealing, where you have a mismatch of this kind, where you have the complete annealing at a 3' end, but the 5' end may not be having any of this match, right? And this is specifically done when you want to introduce some restriction sites into your template, which you want to later use for cloning and for down. So this can be used to add restriction sites to insert, right? So you can modify your insert to have a certain restriction enzyme site, so that once you insert this into your new plasmid, you can also isolate it with using the same restriction sites. And then, of course, you have this one here. This primer will not give you any amplification because there is a requirement to absolute complementarity at the 3' end, because 3' end is where the extension happens with the primer, right? So therefore, this primer will not give you any amplification. So this is something that you need to keep in mind, that we can have some sort of miscomplementation at the 5' end, but the 3' end, the last nucleotides at the 3' end and the last one for sure must be absolutely complementary. Otherwise, this will not give you amplification. And this is also again used to assay for SNPs. So let's say you know that there are 2 variants of this nucleotide in the population. So you could keep one of the complementary nucleotides in this primer. If that nucleotide is present, it will give you amplification, otherwise it will not. And there is a special name for this. Can anybody tell me what is this name? It is called ARMS PCR, Amplification Refractory Mutation System, right? I'm sure you would have read about it. So this is what is PCR. Then, of course, it can be used to check for SNPs. And then, of course, the specificity also depends on what temperature you're using for annealing of the primer. And we'll come to that in a moment. And primer pairs can be standardized for a specific amplification using a gradient PCR. Now we come to the PCR steps. So first, of course, depending on whatever your sample is, you will have the initial denaturation if you're giving human genomic DNA or let's say plant genomic DNA. Then, of course, you have to give a longer time because your initial DNA is extremely huge. For example, human DNA is 3.2 billion base pairs in size. So therefore, you keep it at 94 degrees centigrade for more than 5 to 10 minutes. Then we come to cycles. Cycle denaturation is of shorter time because you are actually denaturing not the whole DNA. Progressively, what would happen is you get amplicons of the size you require. So that is not your entire genomic DNA, but very short. Let's say if you're doing normal TAC, you cannot have fragments more than 2 kb in size. So therefore, here the denaturation time is less. This is your cycle. Cycle has 3 steps. First, the cycle denaturation, which is at 94 at 1 minute. Then you have the annealing, which is where the primer is going to hybridize to the DNA. And this is, again, the range for hybridization is around 55 to 65, depends on the primer composition. And we'll come to it, what is the primer composition and how do you calculate that. And this is roughly around 30 seconds, followed by the final extension where once the primer is now hybridized, it is going to get extended. And this extension happens at around 72 degrees centigrade. And generally, again, there is a consensus of 1 minute for 1 kb. And then, of course, you have the final extension, which is after the cycles are done. On an approximation, you do around 30 to 35 cycles. So if you begin with 1 molecule of DNA, you will end up with 2 raised power 30 or 2 raised power 35 molecules. And generally, you begin with certain molar concentration. So it is always more than 1 molecule of DNA. So X into 2 raised power n is the total number of molecules you're going to get here. And then, of course, you have the final extension of 72 for 10 minutes, after which you can keep your sample at 4 degrees centigrade till infinity. That is until the time you want to use them on the gel, right? So that is your PCR. Once your PCR is over, you would want to load it on the gel and check the size of your amplification using your molecular weight marker, or what is also known as ladder. This is basically a pre-calibrated fragments of DNA, and the concentrations are so maintained that you have some distinct intervals. For example, this is 500 base pair, this is 1000 base pair, this could be 1500 base pair. This depends on which company's ladder you're using. Generally, there are two types of ladder that we have. We have what is 100 base pair ladder. 100 base pair ladder means that the lowest marker here would be 100 bases, 100 base pairs, and then of course it goes up to 1.2 kb, sometimes to 1.5 kb. And then you have a 1 kb ladder where it goes up to 10 kb, and then the lowest is around 1 kb thing. So this again depends on which company's ladder you're using, and also depends on whether you know what is the size of fragment you're going to get, right? Sometimes you may use both the ladders together so that you have, if your fragment is below 1 kb, you still are able to measure the size of the fragment. So this is your PCR now. Now, one of the key properties of PCR, and one of the keys to have a good amplification and the specific amplification, is to design the primers correctly, right? If the primers have not been designed well, you may not get specific amplification. You may actually end up amplifying a region that you're not even targeting. So that is important, that to amplify your, when you design your primers, you take utmost precautions of how you have designed the PCR primers. And this is the experiment that we're going to do today, how to design PCR primers, right? So here you are, we take what are the typical characteristics of the primer that we need to keep in mind. First, of course, is that the melting temperature of the primer must be in the range of 55 to 60 degrees centigrade. In fact, 55 to 65. That is something that you have to keep in mind. A melting temperature of the primer lower than this can cause, non-specific annealing elsewhere in the genome. Then the primer should not dimerize amongst themselves or with respect to forward and reverse primer together. There should not be hairpin loop formation or self-complementarity. We'll discuss each of these points as we go along. And then, of course, there should not be on the genome secondary priming sites. So here you are basically, this is your gene of interest, right? And what are the primer properties that you require that are desirable? First, I mean, if everything goes fine, the best primer is this primer. One at the start of the gene of interest until, let's say, 20 base pairs inside, 20 bases inside, and the other one, which is the reverse primer, again at the 5' primer at the end of the gene and 20 base pair inside. I mean, this would be the best possible combination. You have absolutely your gene of interest getting amplified and nothing else. But the problem is that you have certain properties that needs to be adjusted, right? For example, here, the primer must be unique. So if there is, let's say you're amplifying a region that there is a repeat, then of course you cannot have a primer in this region, right? So this would more be common when you're dealing with non-genic regions of the genome. So you need uniqueness. The primer sequence must appear only once in the template DNA, and the primer must be of 18 to 27 bases. We'll talk of this. Then the primer is working in a pair, so forward and reverse primer together should also have certain properties that you must look into. Primer composition should be around 50 to 60% GC content. So let's say there are only A's here in this area here. So then, of course, you cannot design a primer here. Or there are only T's here, you cannot design a primer here. There are only G's here, or a preponderance of G's, let's say more than 80% G's. Then again, it is not possible to design a primer here, right? So primer composition should also be kept in mind. And also what is important is that whatever GC the primer has, they should come at random. It should not be that the 3' end is absolutely GC rich or the 5' end of the primer is absolutely GC rich. So that is something that you need to keep in mind. Then primer must not be designed to the repeat sequence. So most commonly what we do is we design primers to flanks. If the flanks carry a repeat sequence, then you cannot design a primer here as well. TM must be 55 to 65. So sometimes, and TM is basically a function of the composition of the primer, and composition of the primer is the function of the composition of the template, because blue primer will be the same as what is the sequence here. Likewise, the red primer will be the same as what is the sequence here, and it will, so accordingly, you have to ensure that, this composition of the region that you're going to amplify, again, decides where the primer will be formed, right? Because you have certain set primer conditions of temperature and of length of the primer. You must avoid dimer or hairpin loop formation. We'll come to it in a moment. So here you are. First thing that you require is that your primer should be unique. It should have only one perfect matching site in the genome. It cannot have more than one perfect matching site in the genome. Otherwise, it would be a problem. So that should not happen, right? Then primer length is important. The shorter the primer, the more less specific it is. The longer the primer, the more the chance that it will be a unique sequence occurring only once in the genome, right? So primer length has effects on uniqueness and melting to annealing temperature. The longer the primer, the more the chance that it is unique. The longer the primer, the higher the melting, and this also satisfies your melting and annealing temperature conditions. So the higher the length, the more the melting and annealing temperature, because you have more number of nucleotides coming in. And again, AT has double hydrogen bond, GC has triple hydrogen bond. So higher GC content would also increase the melting temperature again, right? So that is something that you need to keep in mind. Everybody with me, class? You're following? I'm sure you have read all of this. I'm just enumerating these because when we design the primers, we have to keep this in mind. And we will define some basic conditions of the primer, and that we need to that the software has to satisfy before it designs primer to a specific region. So the length of the primer has to be at least 18 bases to ensure uniqueness. And usually the length can be around 18 to 27 bases. Beyond 27, there can be other complications, and that we'll discuss in a moment. This range varies based on if you can find unique primers with appropriate annealing temperature within this range. So here we are. Why should the primer length not be more than 27? Because there are chances that it can form a hairpin and complement itself. For example, you see here, there is a complementation here, there is a complementation here. The other thing is that if the primers are too long, they can find regions of complementarity amongst themselves. So this is a self-dimer, the same primer annealing to itself. And then, of course, now the PCR would happen here. This 3' end will be extended, taking this as template, and this 3' end will be extended, taking this one as template. So effectively, your primers are not going to the PCR or to the targeted template, but actually amplifying each other at the ends, which is not something that you require. Then this could be another case where the forward and reverse primer self-complement. And then again, this gets extended here, and this gets extended here. So these are some of the things that you need to avoid while you're designing your primers. And you also need to keep in mind the composition of both the primers and keep them comparative, so that you do not have enough of these regions, which basically force a certain primer concentration to be out of your actual PCR reaction, right? And what happens is the hairpin formation and self-dimer may not be so much of a problem, because you are doing your annealing at 60 degrees centigrades. So here you are, base composition affects hybridization specificity and melting annealing temperature. Random base composition is preferred. Avoid long AT and long G+C regions. That is important. And usually, as I said, G+C content should be around 50 to 60%. This will give you stability in hybridization. Then, of course, how do you calculate the melting temperature? Melting temperature is generally calculated by a very simple formula. You say Tm equals to 4 into G+C plus 2 into A plus T. So you can very clearly see GC is given a higher weightage because GC has 3 hydrogen bonds and requires higher energy to break down. And AT has 2 hydrogen bonds, so it requires lower energy to break down. So therefore, when you calculate the Tm or the melting temperature, the temperature at which half the DNA strands are single-stranded and half are double-stranded is what is Tm, and this is 4 into G+C plus 2 into A plus T. Also, what you have to remember is there are 2 primers. And in the PCR reaction, you can only give one temperature. So the 2 primers cannot have a Tm that is far apart from each other, right? So not more than 3 degrees centigrade difference should be there between the forward primer and the reverse primer melting temperature. to work in a single PCR temperature. So that is also important, that the two primers must be compatible to each other. So that is something that you do here. And then, of course, annealing temperature is basically a function of melting temperature. And generally, annealing temperature is around about 4 degrees lesser than the melting temperature. So that is something I need to keep in mind. Primer work in pairs, forward primer and reverse primer. So they can be used in the same PCR reaction. It shall be ensured that PCR condition is suitable for both of them. And this is where we look into the temperatures of melting or annealing. The difference between the forward primer and the reverse primer with respect to annealing temperature, or for that matter, melting temperature, cannot be more than 3 degrees centigrade. It cannot be that forward primer has an annealing of 55 and the reverse primer has an annealing of 65. Then they will not be compatible to each other. So the software takes care of this and ensures that the two annealing temperatures of the primer are roughly in the range where they can work together. So here you are. This is your gene of interest, and then you are looking for certain properties of the primer, and because of which you may have to not necessarily design a primer exactly at the start and end of the gene, but you may have to look into the flank and look for a region that satisfies all these criteria of a specific primer design. What is the criteria? One, the primer has to be unique. There should be only one and only one perfectly complementary site in the genome. There cannot be more than one such sites. Second, primer works in pair, forward and reverse. So therefore, their annealing temperatures cannot be too different from each other. Then the primer composition, 50 to 60% GC content must be there for both the primers, because that would provide a stability of annealing. Primers must not be designed to repeat sequence. This is one very important point, because if you look at the human genome, 55% of the genome is repeats. So while retrieving the sequence, you have to ensure that the sequence is retrieved in hard mask, where all nucleotides corresponding to repeats are converted to capital N. That will allow you to design a primer that is not directed to the repeat, because the repeat is not visible in the sequence as such. Then TM is 55-65, compatibility between forward and reverse primers, and avoid dimer and hairpin loop formation. This is what you need to do. So therefore, whenever you're designing primers, what you do is, if X and Y are coordinates of your gene of interest, you retrieve flanks, minimum 500 base pair flanks on the 5' side, and likewise additional 500 base pair flanking sequence on the right hand side. So this is your 5' end, this is the 3' end. So you start with X minus 500 if X is the starting coordinate of the gene, and you retrieve until Y plus 500. So effectively, you are retrieving 1000 base pair extra, 500 base pair at the 5' end, 500 base pair at the 3' end. Everybody with me, class? You're following?
Yes, yes, sir.
Yes, sir.
Right. Okay, good. So what you're doing is, this is your gene of interest. So the ideal situation is that you could directly design, you could just read the sequence here and make a 20 base pair primer here and 20 base pair primer here. But that ideality cannot work, because the primer has also to satisfy certain basic minimum conditions of TM, of length, of specificity. And therefore, what you do is, whenever you're designing a primer to a particular target, you first retrieve the sequence with certain flexibility of flank. So you have, let's say here, 500 base pair extra on the top, 500 base pair extra on the bottom, right? Then you retrieve sequence with extra flanking sequence. Then what is also important is that you retrieve the sequence in hard mask. So if there is anything that is corresponding to a repeat sequence here, that would automatically be converted to N. So therefore, the software where you're giving your sequence as input for designing the primers would know that primer cannot be designed here. So therefore, it is not those areas, while they're part of your reference sequence that you have, they will be excluded from primer designing by the software, because it knows that if there is an N, it is not seeing the nucleotide there. So therefore, it cannot basically make a primer to those regions. So retrieve sequence in hard mask, repeat nucleotides converted to capital N, ensures that primers are not designed to repeat, which is something very important. Because as I said yesterday, if you look into the human genome, there are 1.5 million copies of an ALU sequence, right? So if you mistakenly design one of your primers to an ALU sequence, you are effectively sending your primer to 1.5 million different sites in the genome. And then in that case, you are amplifying a lot of nonsense along with the sense, and sense will be so masked that you will not be able to see what is your actual corresponding fragment. So therefore, you need to mask the sequence. If the gene is on the plus strand, retrieve the sequence directly. I've already told you. If it is on the minus strand, then you have to do a reverse complementing so that, later on there is no confusion. So with that background, let's now get back to designing the primers. And our task today is to design primers to amplify gap DHG, right? So because we've already done gap DH once, in terms of retrieval, we'll take that forward and we'll retrieve the sequence of gap DH. So here we are. We go to, again, UCC genome browser. So here we are in UCC genome browser, and we go to UCC genome browser home. And from here, we go to genome browser here. This is the first link, right? And we could click here. This will take you to the home page of the genome browser. It gives you the two mirror sites. We select one of the two mirror sites. I select the Asia site. So I'm here. And now I'm on the home page of the genome browsers, right? And as I told you the first day, a whole lot of sequences are lined up in this particular browser here. On top, by default, is the human reference genome. And within the human genome, reference human genome, the default assembly is the latest assembly, that is the GRCH38 assembly, right? That is by default. Excuse me. And if you want to go to some other organisms, there are a whole lot of organism list available here, human, chimp, bonobo, and so on and so forth. And almost all model organism that we use today is available. They're also available snake, lizard. Then, of course, you have the frog and Tibetan frog. And then, of course, you have Drosophila, you have C. elegans, which is the flatworm. You have Apis mellifera, which is your honeybee, right? Then Drosophila, and then, of course, Saccharomyces cerevisiae, and Ebola virus, SARS coronavirus, and monkey virus 2, right? So these are some of these. And yesterday, at somebody has asked, so there is another UCC genome browser for prokaryotes. You can have a look at that as well. Most commonly used one is this one. You want to pull out the sequence of GAPDH gene. So if you know the position, you can directly write the position. Else, I'll write GAPDH, GAPDH, DH, and I'll wait for it to prompt. Once it prompts, I'll select my gene of interest, GAPDH, and I say go, right? It has now changed the locus. If you see, it is now on chromosome 12 and position 653. This one here, this is position 6534, 517, 2, 6538, 371. What you need to note is the size of the gene. This will be required for our understanding of what we're doing. What we have now is the location of the gene. We also have the size of the gene, right? We go to view and we go to DNA. And because we're designing primers to amplify the full gene, the primers must be outside the gene. So we will retrieve extra 500 sequence at the 5' end and another extra 500 sequence at the 3' end. So effectively, we are retrieving 4855 base pairs of sequence, right? Everybody with me, class? You follow?
Yes, sir.
Yes, sir.
Okay. So this is what we are doing. We are adding extra 500 base pair at the 5' end and another extra 500 base pair at the 3' end, because we still do not know what is the composition of the flanks, right? It could be all repeats. If it is all repeats, we'll have to further extend our search and extend and basically pull out another bit of extra sequence. But let's see how we go about it. So now we come to the sequence formatting option. And here what we'll do is, compulsively what we'll do is, we'll do a mask repeat, and we mask the repeats to capital N, right? Because we don't want our primer to be designed to a repeat sequence. So what we do is we make the software that we're going to use blind to the repeat sequence. So we convert all the repeat sequence corresponding nucleotides to capital N. The software will know, okay, there is some sequence here, but I'm not supposed to design a primer in this region, right? Okay. So, and then because our gene is on the plus strand, if you go back here, you can see the arrows are towards the right hand side. Can you see that? So which means that the gene is on the plus strand. So therefore, we don't need to do a reverse complementation. So we go back again, we go to view, we go to DNA, and we go to our 500 base pair extra on the 5' end and 500 base pair extra on the 3' end. And all uppercase is what we want. We want the repeats to be masked to capital N. To get the mask sequence, you have to check this box first. When you check this box, then only you're exercising your mask repeats option. And we're doing a hard mask where the sequence of the repeat is not visible, although it is present as capital N. So you know that this region is the repeat, but it is not visible to you in terms of the nucleotides that are there in it. And then we say get DNA because our gene is in the plus strand, so therefore no problem of doing a reverse complementation. So if you see here, this is now your FASTA format sequence. You have greater than sign, you have AG38 DNA, you have range chromosome 12, you have the start coordinate, end coordinate. This will now accommodate for the 500 base pair extra you have started at the 5' end and 500 base pair extra that you're retrieving at the 3' end. So this coordinate is 500 base pair above the start of the GAPDH gene, and this one has 500 base pair below, extra to the end of the GAPDH gene, right? If you can see it, it mentions 5' pair equals to 500, 3' pair equals to 500, which means there are 500 base pair extra nucleotides on either end. And then you have the strand is plus because you have retrieved in the plus strand, and repeat masking is 2N, capital N, right? So which means that you're repeat masking here. So at the top, there is not much of a repeat, but if you scroll down further down, you will see you have some repeat sequence in the flank towards the 3' end of the sequence. Can you see that, class, everyone?
Yes, sir.
Yes, sir.
All right. So, okay. So this is now our template. We are going to design primers to this, right? So let me just select this and cancel. This is going to print. So we can select this here and keep it as our, in the buffer. So we say copy, right? Now we move to the primer designing tool. This is known as Primer 3. Primer 3 is the most common primer designing tool that is used for primer designing, right? So you on primer 3 dot input, right? So we can click here, and this takes us to the actual primer 3 web version 4.1.0 pick primers for a DNA sequence. And you can see here the pick left primer and pick right primer option is clicked by default, right? So because by default, this is a tool for designing primers. So this option is activated by default. But in case you're making a nucleic acid hybridization probe, then you can check on this. All right. So let me one more time take you here and explain what we're going to do, right? So here you are, and let me try something different. So what we have done till now is this. So what we've done is, this is our gene of interest. This is your GAPDH, right? This is the length of this is, if you see here, this is 3, 8, 5, and 5 base pairs, right? This is the length of this gene that we have here, right? We started our sequence here. So this is our first coordinate, right? And this is our 4, 8, 5, 5. Remember, we have 500 base pair extra at the 3' end, 5' end, and again 500 base pair extra at the 3' end. So if you look at the total sequence that we have isolated, we have isolated this sequence from here until here. Everybody with me, class? Right? This is the sequence that we have, which includes in the center 385 base pairs of the gene of interest. So first thing that we want to tell the software is that do not design a primer internal to the gene of interest, right? So what we do is we use an option to tell the software that primer is not to be designed inside the gene, right? So what we use is what is known as excluded region option. So when you mention the excluded region option, you are saying, okay, this is the region where the primer is not to be designed. So if you want to amplify the full gene, the primer has to be outside of the gene, not inside the gene. Everybody with me, class? You follow?
Yes,
Yes,
sir.
sir.
Okay. So this is where we are. What we have is the gene sequence, which is 3855 base pairs in the center of the gene sequence or GAPDH gene sequence, and you have 500 base pair extra on the 5' flank, and you have 300 base pair, 500 base pair extra on the 3' flank. And what we want to now tell the software is that do not design primer to this region here, right? So for that, what we use is what is known as the excluded region option. Now, how do you write the excluded region option? You have to say where is the excluded region starting. So with reference to this, this will be x will be, what is the coordinate of x now, class? If this is 1, this will be 501, right? Okay. What you have retrieved is a sequence that contains your gene of interest sequence. Along with it, it also has a 500 base pair extra 5' end and a 500 base pair extra at the 3' end. So if you look at this whole genome, whole sequence that you've isolated, your gene is starting at 501 because 500 is your 5' flank sequence, right? And you want to mention what is the excluded region. So how do you mention the excluded region? You have to mention the start from where you want to exclude and until what length do you want to exclude, right? So you want to exclude from 501 until the next 3855 bases, which is the size of your gene of interest, right? So we'll exercise this option once and then we'll come back here and we'll talk more. So here, first, this is the option where you can paste your sequence. So we'll paste our sequence here, Control V, right? So this is our sequence in FASTA format that we're pasting here. This is the sequence we're using for designing our primers, right? Of which this is the sequence that contains your in the center contains the 3855 base pairs of your gene and 5' end 500 bases extra, and at the 3' end another 500 bases extra, right? We are picking primers for our use. So pick left primer, pick right primer. This will be by default on. First, what we define is what is the region that is to be excluded. How do you define that? For example, here if you say 401,7, forbid selection of primers in the 7 bases starting from 401. So if you do not want selection of primers 7 bases downstream of 401, you have to mention 401,7. What do we want? We do not want picking up of primers from 501 until the next 3855 bases, which is the size of your gene of interest, right? So we'll say excluded region 501, 3855. So here we are. So I take it, unless you ask me a question, I will take it that by default you're following. So first what we're doing is we're telling the software that what is the region where the primer is not to be designed. Obviously, if you want to amplify the full gene, the primer cannot be inside the gene. So you're saying, okay, exclude from 501 until the next 3855 base pairs, which is the length of the gene. You cannot design primers. Now, the problem with this is, let me just go back to the PPT again. I have told the software, do not design primer in this region, right? So we have clearly told the software, primer cannot be in this region. Mute, please. So here, yeah, so you basically are telling the software not to design primer here. But what the software can do is it can design both your primers here, right? One primer here, one primer here, right? Because it has no other instruction other than not designing the primer here. Or what can also do is one primer here and one primer here, which is not even within the target, right? So effectively what would happen is you could only be amplifying the flank instead of amplifying the gene. So you must have an option of telling the software that, okay, you need one primer here and the other primer here, right? One primer on the left flank, the other primer on the right flank. Everybody with me, class?
Yes.
Right?
Yes, sir.
How do you tell that?
Yes, sir.
One way of telling that would be you define your product size range, right? You are saying, okay, my product size range is this. If both the primers were to be designed to the left flank, the maximum product size would be anyone. If you have both the primers getting designed to 500, right? That is your maximum size of the flank. So it cannot be more than 500. So the moment I say my product size range is 3855 to, let's say I give 200 base pairs of flanking sequence flexibility. So I'm effectively telling them not to design both primers on the same flank, right? So what I'll say is product size 3855, which is the size of my gene. So it cannot be any less than this if I'm amplifying the full gene. And 2, I'll give them a flexibility of, let's say, 200 bases. I'll say 4055. Why am I giving this flexibility? Because I need primers that satisfy my base conditions. What are my base conditions? TM of how much, class? 55 to 65. Everybody with me, class? You follow?
Yes,
Right?
sir.
Yes, sir.
I'm also defining my primer length. I want a specific primer. So there is a minimum and maximum to it. So I'm saying primer length cannot be less than 18 bases and should ideally not be more than 27 bases, right? So these are my conditions. I'm also saying my GC content should be for the primer should be around 50 to 60%. So given these conditions, I will have to give the flexibility to the software to scan through the flank to find a region where a primer can be designed with 18 to 27 base pair length that also satisfies the TM and also satisfies my GC conditions. Everybody with me, class? So therefore, you have to give certain flexibility. Otherwise, it is not primer designing. You can simply take the sequence from here, and where is my, okay, it is here. So you can simply take a sequence here and say, okay, 20 bases is the forward primer. This one is another 20 bases, the reverse primer. But because the primer is also having certain conditions, you have to, and all these conditions are basically a function of the composition of the DNA here, the sequence that you have here. So therefore, you have to give a flexibility of a little so as to, find a region in the flank that satisfies all these conditions. Everybody with me, class?
Yes, sir.
Okay, so some of you are following closely. Okay, so what we do is we go back here. First, I'll show you that if I'm not giving the option of any other option except that specifying that, okay, do not design primer within the gene sequence, which is starting at 501 and until the next created 55 base pairs, what is the primer it is going to pick up, right? So we are just going by random thing, and that is to show you that this will be a wrong primer design experiment. So let's say I can, for example, I can also show you what are the primer picking conditions that the software has by default, right? So if you see here, primer size 18, optimum 20, maximum 23. So optimum is the best fit. It will first look for the best fit primer. If the flanking sequence does not have enough compositional difference that it allows for the best fit primer, it will look for the the maximum minimum ranges as well, right? It will first try to find a primer with optimum primer size of 20, primer TM of 59, and a GC content of 50%. If not, then it will also vary its range because you have to give certain flexibility to the software because this is totally dependent on the composition of the flank, right, class? Then there are other options. This you can look for how much 3 times self-complementarity. Thing for everything there is a help file available. You can look for the details, and then of course what you say is you can say now pick primers. Remember, right now we have not given anything except that saying okay, do not pick primers for the region where my gene sequences, right? So I say pick primers, and these primer sets will be wrong. Now, if you see here, how do you see the results? If you see, it gives you a primer pair here, right? Left primer, right primer. The start of the primer is at 153 bases. Your gene starts here. This X is your excluded region. So this is your gene start. So your starting primer is here. Likewise, your reverse primer is at 350, which is again within the flank itself. So what you get as an amplicon would be just this much, which is not even a part of your gene. So that is why you need to also give the other option of very smartly telling the software that, look, I want one primer on the left flank and I want the other primer on the right flank. Everybody with me, class? You follow?
Yes, sir.
Yes, sir.
Right. Okay. So I hear 3 distinct voices and sometimes a male voice. And we have some 40 participants. Many of them prefer to keep quiet, which is fine. So this is not the correct primer design. So what we do is we go back here and we write the next option so that we ensure that one primer gets picked up at the left flank and the other primer gets picked up at the right flank. How do we do that? We will say, so here you are, you have this product size option, right? So we can say the product size range for us is 3855, which is the size of my gene. If I'm amplifying the entire gene, I must have a 3855 base pair amplicon, right? And then because I want to give some flexibility in the flank where it is going to search for the primer that satisfies these conditions, I'll also mention that, okay, until 4055. So initially I'm giving a 200 base pair stretch so that it is able to find the primer in this region. Then I'll see based on the results, I can refine this product size range to slightly lesser so that my primers come closer to the target, which is supposed to be amplified. So I'm giving 3855 to 4055, right? And then again, I say pick primers now again one more time. So here we are. We say pick primers. Everything else is left to default. We are not changing anything here. We pick primers now. And now if you see, this is your front primer, right? This is your forward primer. And you can see it is before the start of your gene, slightly upstream of the gene. So we'll try to fix this now. But before that, let me show you the other primer as well. So this is your excluded region of the start of the gene. This is 501 until the next 3855 base pair. So if you scroll down, this is until the next 3855 base pairs, right? So here you are. This is where the gene ends. And you can see the reverse primer is close enough to the gene end. So probably this primer cannot be moved any further inside. But the forward primer, if you see here, this is quite far apart from this. So if you see here, so basically, if you're looking at this is clearly 60 bases. This is another 10, 15 bases at least, right? So 60 plus 15 is 75. And here again, you have close to almost 40 bases extra. So 100 bases away is your primer from the actual gene start. And you can do that also. This is 361. If you see here on the top, everyone, the left primer begins at 356. The length is 20, which means it ends at 356 plus 20, 376, right? And your actual sequence of the gene begins at
501.
501. Very good. Thank you very much. So you know that from 376 to 501, what is the number of nucleotides extra? So what you want ideally is to bring this primer closer. You don't want extra nucleotides to be incorporated into your amplicon. So we'll try to do that by reducing the amplicon size, right? So what we'll do is we had given the option of 3855 to 40455. We will now make it 3855 to 3955. So we are giving a very minimal stretch where it can scan and use these properties of the primer and see if it satisfies these primer properties and give us a primer pair. Sometimes this will work, sometimes it may not work. If it doesn't work, then of course there's nothing that you can do. You'll have to amplify it the way you amplified with slightly distant primers, and then use restriction digestion to digest to the nearest target size that you can get, right? So here we are. We reduce it to 3855 to 3955. And then again, we go to pick primers and we pick primers again. And the first thing we're going to see is whether our forward primer moves inside or not. And if you can see here now, it is much closer to the gene start. Can you see that, everyone?
Yes, sir.
Yes. So I guess this is the best that you can get. There are only some 10 extra nucleotides. You cannot move any closer by reducing the range, because probably this is the region that does not satisfy. The composition of this region does not allow you to create a primer that is satisfying your basic primer picking conditions. So therefore, we can go ahead with this primer pair and do an amplification. Remember also, it will give you another 4 set of primers at the down. So here we are. You have some extra primers, right? If you have one more and you have another more here, right? So this one is, so it has only one more extra pair, which is basically roughly the same as the initial primer, just that it is a bit of a nucleotide one here and there. If you can see, this is starting at 462, and it is 23 base pairs, 23 bases for the forward primer. So we can just have a comparison from the top. So this is starting at 466, right? So that's the only thing. And this is 23 bases, that is 20. This is 22 bases, that is 23 bases. Generally, the best scoring primer comes at the top, so you can go by the best scoring primer. This is the primer pair that you need to order through your oligosynthesizer. Once these primer come to lab, these will be in lyophilized state. You then, dilute them to the concentration, keep a stock, and then also dilute them to the final concentration that you want for your PCR reaction. And then first you have to standardize the PCR. What is standardization? You have to find the right TM for the primer at which it gives you a specific amplification. For that, you do a gradient PCR, and then of course, find the temp. So gradient PCR essentially is when in one PCR, you can vary the temperature of individual lanes of your machine, right? And that allows you to determine which temperature is best for your primer pair, where you have high amplification and no non-specificity, right? Sometimes you may also have to do a magnesium chloride standardization. Generally, 1.5 millimolar concentration is fine, but at times you may have to do a bit of a MgCl2 concentration standardization also. You may also have to use some adjuvants like bovine serum albumin, sometimes DMSO and other things, so that your primer pair works. So This is the precaution that you can take in dry lab. But of course, when it comes to wet lab, things get slightly different. Your primer may work in the first attempt itself. Sometimes you may take time to standardize the primer and find the best optimal conditions for that primer in the wet lab, which may be slightly different from what you predicted in the dry lab. So this is your primer designing. I hope I have given you a flavor of how to go about doing it the correct way.